Hello students, in this part we will discuss on structure of DNA. Structure of DNA. As we take the small background history of DNA, DNA was firstly discovered by Frederick Miescher. DNA was discovered by Frederick Miescher in 1869, and in 1889 it was proposed that it is nucleic acid by the Altman. Altman proposed nucleic acid in 1889. The term DNA term was proposed by Jacques. In this part we will study about the structure of DNA, how the DNA is present in the cell. DNA structure was proposed by J.D. Watson and Crick. The structure of DNA was proposed by Watson and Crick in 1953. They proposed that DNA is present in double helix structure. Double helix structure means that the stair which is in spiral stair कह सकते, spiral railing कह सकते हैं. Watson Crick proposed double helix structure, or you can say that the double helix model was proposed by Watson and Crick in 1953. What were the features of DNA double helix structures and how they interpret from the double helix structures? The features of double helix structure was DNA is polymer of nucleotides means the compound which is made up of pentose sugar and pentose sugar, which one? Deoxyribose. Pentose sugar with the nitrogen base and phosphoric acid, that forms the nucleotide. A little bit we will draw the structure of nucleotide. It means pentose sugar. In pentose sugar there are a ring which is made up of 1 oxygen and 4 carbon atoms. The oxygen atoms, and these are 4 carbon atoms, and 5th carbon remains as a R. This is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and 5th carbon is in the form of R. At carbon number 1, nitrogen base is attached. The bond which is formed with the attachment of nitrogen bond and nitrogen base, that is called glycosidic bond. And phosphoric acid is attached at the carbon number 5th carbon number is here. Phosphorus is attached at the carbon number 5th, and the bond which is formed with the attachment of phosphoric acid, that is called phosphate, that is called phosphodiester bond. Phosphodiester bond. Clear? This is the structure of nucleotide. So first, DNA is made up of DNA is polymer of a nucleotide. The first feature which was proposed by J.D. Watson was DNA is a double-stranded, or you can say that DNA is double-strand. DNA contains double strands. DNA के अंदर 2 strand होता है. You can say that DS stand for DS double strand, SS single strand. But they propose that DNA is a double stranded, या DNA contain double strand. Second one, both the strands are anti-parallel to each other. Both the strands are anti-parallel to each other. As we know that DNA is made up of DNA is a polymer of nucleotide. Watson and Crick proposed the double helix structure means spiral stair, spiral railing. DNA contains double stranded of nucleotide, and second one is both the strands are anti-parallel to each other. How these both strands are anti-parallel to each other? If one strand contains the polarity 3' to 5, then the other one is 5 to 3. How these anti-parallel strands are generated? Suppose this is we can take the one nucleotide. This one is the one nucleotide and it contains carbon number first, carbon number 2, carbon number 3, carbon number 4, and fifth carbon is here in the form of R. Third carbon, as we know that DNA is the deoxy. Why it is called deoxy? Because at the second position oxygen is reduced. Here it contains H and H, and third at H and OH. Nitrogen base is attached at carbon number 1. Carbon number 1. We will take one more nucleotide. Suppose this is the another nucleotide of DNA. Carbon number first with attached the nitrogen base, carbon number 2, carbon number 3, carbon number 4, and fifth carbon remains here. Carbon number 5. At fifth position, phosphoric acid is attached or phosphate is attached with the fifth position, and here is also the bond which is formed between the fifth carbon, or we can say that at the phosphoric acid. Phosphoric acid attached with at the fifth position of the carbon, and bond which is formed, this bond when one nucleotide attached with the other at the fifth position, this bond is known as phosphodiester bond. Phosphodiester bond. And carbon number 5, the nitrogen base is attached, that is called glycosidic bond. Clear? Here we can draw the structure of one strand. One strand means this is here carbon number 5, fifth carbon is free here, and this one it contains carbon number 3. If we join a line, then we will receive, or we can take the diagram such like as one strand. This one, it has polarity at the fifth carbon present here, fifth, and this one is the third, free 5' and 3'. This is the one strand, and we know that both the strands are anti-parallel. Anti-parallel means if one contains the polarity 5' to 3 at fifth carbon in place, or you can say that in front of fifth carbon free, the second strand contains number of 3 carbon free. So we can take one more structure, but at that time we have seen that both the strands are anti-parallel to each other. Then how these nucleotides will be taken? Just opposite. To draw this structure, this one. Carbon number 5th, first, 2, 3rd, 4th, and this one is carbon number 5th. In front of carbon number 1, we can take carbon number 1. So that is glycosidic bond is formed with attached this one. And second, we draw the structure of this one. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this one is carbon number 5. Here is first, second, third, fourth, fifth. Third, it contains OH. Here is P, and it also contains OH, CS2OH. Here is also bond formed, that is called phosphodiester bond. We'll take the structure. This arm contains 5', fifth carbon free here. In front of it, it contains carbon number 3rd free here, just opposite. And here the carbon number 3 free here, and in front of it, it contains carbon number 5th. So we draw the structure of the spine. 2 strands, and both the strands are anti-parallel to each other. How this structure can be seen? We have seen that the both strands are twisted. दोनों जो स्ट्रैंड है ये ट्विस्टेड एक प्रकार से लिपटे हुए होते हैं, जस्ट इनटू द क्लॉकवाइज मैनर, मींस राइट हैंडेड. We can take the example of a spiral stair. Spiral stair means the stair which starts from here and at first floor it goes up. Both the strands twisted right-handed, or you can say that clockwise in a spiral stair manner or a pattern. They have two strands, and though both the strands are anti-polarity or anti-parallel to each other, both strands are twisted clockwise, means right-handed in a spiral stair manner. First we draw the structure of spiral stair. It contains two strands. This one like manner, this is the first strand, second one here. Second here. These are twisted in such like as manner, or you can say that in a clockwise manner. If one strand contains the polarity 5 prime, then this one contains 3 prime. This is the one strand. We have seen that they have both two strands and both the strands are anti-parallel. Anti-parallel means if this strand contains 5th carbon free here, then the opposition is 3 carbon free here. Here is 3 carbon, the opposite is 5 carbon free. Clear? This is present in just a spiral stair like structure. Both the sides are not cross interconnected or cross with each other. Both are run parallel to each other. Fourth one is DNA contains nitrogen base. DNA contains nitrogen bases, and there are two types of nitrogen bases: purine and pyrimidine. Purine and pyrimidine. In DNA, purine is to adenine and guanine. And pyrimidine, as we know that pyrimidine is 3 types, but DNA it is used only cytosine and thymine. In DNA, purine and pyrimidine form attached with each other in double hydrogen bond and triple hydrogen bond, so that the diameter is not affected. Diameter remains same of the DNA. How these purine and pyrimidine are attached with each other? Adenine bonded with thymine with a 2 hydrogen bond. Not a, this sign is not for it is equal to. Adenine bonded, it forms 2 hydrogen bond with the thymine, and guanine attached with cytosine with triple hydrogen bond. In such manner, DNA pure nitrogen base are present. Clear? Next feature one. If we draw a imaginary line means centrally line, we can pass it. We show that we can take the distance of diameter. Diameter means the length from outside to inside. This is known as diameter. The diameter of DNA is 34 angstrom. Sorry, not 34, 20 angstrom. The next feature was the diameter of DNA is 20 angstrom or we can say that it is nearly 2.0 nanometer. This is the diameter from outside to inside. This is the diameter of the DNA, and DNA diameter is 20 angstrom or you can also write 2.0 nanometer. We have seen that if we stand here, then this is the long, this one here, this is the long deeper depression, and this one is shallow depression. Then one turn means when we go in a spiral stair from the ground floor to upper floor, we can rotate at 360 angle. This one rotate or one turn at 360 angle is known as a pitch. Number 6 there, the one turn of the DNA, one turn of DNA is called a pitch. What is pitch? If we start to climb up a spiral stair from the ground floor and then this moves here, this one like, and reach at the top floor, just above the ground floor at the top floor at the same point, then we can rotate 360 angle. This 360 angle one turn is known as a pitch. The distance of one turn is 34 angstrom or we can write up is 3.4 nanometer. How this one turn or one pitch is arises? Suppose we are standing here. This is the deep depression, this one is shallow depression, and depression is known as a grooves. So if we measure the distance from here, one deeper depression, shallow, and one deeper depression, means deeper to deeper depression, consecutive two consecutive deeper to deeper depression, or two consecutive shallow to shallow depression. This one is deeper depression is known as grooves, and this one is shallow grooves. The two consecutive shallow to shallow or deeper to deeper distance is known as turn. One turn, and one turn is also called pitch. This distance means deeper to deeper, grooves to deeper grooves. This distance is known as a pitch or one turn. You can also say that shallow to shallow. We count shallow to shallow. This is the one pitch, and this one pitch or one turn contains 10 nucleotide. Next feature is one turn contain 10 nitrogen base, or you can say that 10 base pair. Nitrogen base pair means cell deeper to deeper. If we count here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. One turn means one pitch contain 10 base pair. So the distance between two consecutive base pair is, if the one turn contains 34 angstrom. 34 angstrom, that means 3.4 nanometer. One turn length. The next point is the distance between two base pair. 10 बेस पेयर के बीच की डिस्टेंस कितनी है? 3.4 नैनोमीटर. 2 बेस पेयर की डिवाइडेड 3.4 मीन्स 34/10. And how we calculate the distance between two consecutive? One turn contain 10 base pair, so we can also divide it with 10. So 0.34 nanometer. The distance between two base pair is 0.34 nanometer, or we can also write it 3.4 angstrom. This is the 34 angstrom, which is the distance of one turn, and one turn contains 10 base pair, 10 nitrogen base pair. and the consecutive distance of two base pair consecutive distance is known as that is equal to 0.34 nanometer, or we can also write it 3.4 angstrom. This model was proposed by J.D. Watson and Crick in 1953, and they called it double helix model. As this is the DNA, as well as we know that and there is 5 types of DNA: DNA A, DNA B, DNA C, DNA D, and DNA Z. How these DNA is present and how many base pair present in one turn or one pitch, then those are different. We will study about different types of DNA and next process in the next part. In this part we will discuss only the structure of DNA, the structure of DNA which was proposed by Watson and Crick in 1953. This model is known as double helix structure.